Good morning, everyone. I hope everyone has had a good few days here in Rome. Today, I wanted to talk about our potential therapeutics for chronic diseases and chronic inflammation and chronic inflammation-mediated diseases. So, some forward-looking statements for a public company. The immune system can be described as a pendulum. With the tendency in healthy individuals and a healthy immune system to always be towards the homeostasis. That is typical. However, 1 in 3 individuals globally have a tendency of their immune system being aiming towards the inflammation state and being stuck there. And that causes a significant number of diseases involving autoimmune disorders, cardiovascular disease, GI diseases, hepatic diseases, and renal diseases. The cause of chronic inflammation is not well known, but it tends to have multiple causes, as we've seen. So just some staggering statistics for chronic inflammation and chronic inflammation-mediated diseases. 74%, about 43 million of all deaths was in 2021 attributed to chronic disease and chronic inflammation-caused diseases. Globally, as I said, 1 in 3 individuals suffer from chronic inflammation. 129 million people in the US suffer from at least one chronic disease or chronic inflammation-mediated disease, with 43% having more than one. And the same percentage is true in Europe as well. And the cost of treating these chronic diseases is expected to reach about $47 trillion worldwide by 2030. So staggering impact and greatest cause of morbidity and mortality in the world. So looking at the top selling drugs worldwide, if you look at these top selling drugs, you'll notice that with the exception of the GLP-1 receptor agonists, all the other ones are immune modulators. Either they activate the immune system or deplete the immune system, and that has caused a different modality when you're looking at the immune system, and that being immune cell depletion or overactivation. And as we've seen with a lot of these monoclonal antibodies, as well as inhibitors that inhibit the immune system or an aspect of the immune system, these depletions do have significant adverse side effects that have been noted in many of the commercials you've seen for many of these products, which involve significant risk of viral, bacterial, fungal infection, but also significant cardiovascular impacts and renal impacts and hepatic impacts that are also noted as adverse side effects with multiple monoclonal antibodies as well as the inhibitors that are used to modulate the immune system. As a company, we are targeting a cell-based methodology for modulating the immune system with the aim of approaching homeostasis and maintaining homeostasis, which would allow your body's natural response to regenerate tissue, to heal itself. And what I will do today is I will go through 3 of our product candidates and show you some of the data from animal model studies as well as one clinical trial to show you that it does work. It is possible to do that using fibroblasts. So our product candidate pipeline, the ones that I will discuss today are CYWC628, which is our product candidate for the treatment of diabetic foot ulcer, a chronic disease. CYMS101 is for the treatment of multiple sclerosis, an autoimmune disorder, again, another inflammation-based disease. And then Cybrosal, which is our product candidate for the treatment of degenerative disc disease. I won't go through that today in the interest of time. And then CYPS317, which is our product candidate for the treatment of psoriasis. Now, we'll start with the product candidate for diabetic foot ulcer treatment. And just to give you some statistics on diabetic foot ulcers, 6.3% of adults with diabetes will develop a DFU. 20% will require lower extremity amputations, of which 10% will die within the first year, 40% recurrence rate within the first year, and 70% recurrence rate within 3 years. They have an incredibly high rate of infection, about 60% infection rate, and an increased rate of mortality of about 50 to 68%. Again, diabetic foot ulcer is a chronic wound that's stuck in the inflammation phase of the wound healing and do not progress into the proliferation and remodeling phase. And that's true also for venous leg ulcers, bed ulcers, etc. So why have we chosen fibroblasts to treat diabetic foot ulcers? The reason is very simple. Fibroblasts are involved in every single stage of the wound healing process, from hemostasis to inflammation to proliferation and remodeling. And as I mentioned, in the chronic wounds, the wounds are stuck in the inflammatory phase and do not proceed to the proliferative phase. And that's because the inflammatory cytokines, growth factors, and the impact on inflammation and locally and systemically prevent the wounds from healing. So here's what our product candidate looks like. What we do is we utilize spheroids of fibroblasts instead of using single-cell approach. These are topically administered for diabetic foot ulcer treatment. And as you can see on the left panel, those are the spheroids on the surface of a chemically induced chronic wound on a diabetic mouse. And you see within 6 hours, these cells migrate onto the surface of the wound and release all the necessary chemokines and cytokines and growth factors that are necessary to initiate the natural wound healing process and push the chronic wounds through the proliferation and remodeling phase. So, and these cells do not graft. They go away within about 4 to 7 days completely, and we have data to illustrate that as well. They're not meant to graft at all. So looking at some of the data, and we've compared this against graphics from Smith & Nephew, which is a FDA-approved product for the treatment of diabetic foot ulcer. As you can see at day 19 in the chronically induced wounds on the back of diabetic mice, at day 19, we achieved an 83.8% full closure of the wounds as compared to about 66% with graphics and 51% for the standard of care treatment for control. Now, in the interest of time, I won't show you all the data for this aspect of it, but it's not just important to close diabetic wounds or chronic wounds in an accelerated manner. It's also important to be able to close them in a quality manner so that they do not recur. So what we have done is we have assessed the quality of the wound healing with our treatment, and we have significant improvements have been demonstrated for reepithelialization, granulation, cell proliferation, neovascularization, which is really important in maintaining a good quality wound, as well as fibroblast recruitment and proliferation and keratinocyte migration and epithelial to mesenchymal transition, which are all aspects of good quality wound healing. And we have been able to show that it is significantly better than the FDA-approved product. And if anyone's interested, I can surely share that data with you. So in summary, we've been able to show that spheroids do attach and migrate on the surface of the wound and release the necessary chemokines. We've been able to show a 58.5% reduction in wound size within 4 days after treatment with a single treatment, and 83.8% wound closure within 19 days as compared to the FDA-approved product, and significantly improved quality of the wound healing. Again, this methodology or this method of treating diabetic foot ulcers or chronic wounds is being able to return the tissue and serum inflammation back to a homeostasis level so that it could naturally heal. Next, multiple sclerosis, an autoimmune disorder, about 2.8 million people suffering worldwide, and the MS prevalence has increased significantly since 2013, about 1 million diagnosed in the US, about similar in Europe as well, with a prevalence rate of about 375 out of 100,000. Again, it's an autoimmune-mediated chronic inflammation-based disorder. And we have utilized 2 different mouse models and rat models to study the impact of treatment with our product for multiple sclerosis. And as you can see in the first one, this is in the rat EAE model. And as you can see, with a single treatment, we were able to improve the EAE scores without a relapse. As you can see with adipose-derived stem cells and control, you get relapses through the whole period, and we were able to follow this for 16 days 60 days without any relapse with a single treatment of our product. And this was just basically 300 spheroids containing about 1 million cells total, a single intravenous administration. And as well, we looked at the impact on the immune system systemically, and as you can see here, in this data looking at in vitro study on the left side and in vivo studies on the right, we were able to significantly improve T regulatory cells expansion both in vitro and in vivo, but at the same time, significant suppression of reactive T cells, Th17 cells, and IL-17 expression, both in vitro and in vivo, with a single administration of these spheroids. We were able also to show significant upregulation of anti-inflammatory cytokines and programmed death ligands, and significant downregulation of key surface markers such as CD40, CD80, CD86, responsible for inflammation, and as well significant inhibitor of dendritic cell maturations were noted as compared to both adipose-derived stem cells and bone marrow-derived stem cells. We were also able to show in this study a significant increase in expression of myelin. That hasn't been shown as compared to control and adipose-derived stem cells. This is in the cerebral cortex of EAE mouse model. We were also able to show, additionally, using secondary assays such as pCNA, that it does indeed increase the expression or proliferation of oligodendrocytes, which are responsible for the creation and secretion of oligodendrocytes, myelin sheath, excuse me. We carried out a second animal model to assess and confirm remyelination. This is the cuprizone model, which is very used routinely in MS work and remyelination. As you can see here, both at single administration and multiple administration, at 7 weeks and 9 weeks, we see significant expression of myelin. So this is something that we were able to confirm again using another animal model, that we do indeed remyelinate, and it does remyelinate significantly, which is very important in the treatment of multiple sclerosis, but also other neurodegenerative disorders that lead to myelin destruction. So we were also able to show that microglia, which are responsible for the destruction of the myelin sheath, we were able to significantly reduce their expansion in the brain, in EAA rat brains. So in summary, we have been able to show significant suppression of reactive T cells, significant expression of expansion of T regulatory cells, as well as significant increase in remyelination, which has not been published by anyone else at this point using a cell-based therapeutic. Next product is for psoriasis, CYPS317 for the treatment of psoriasis. 123 million people worldwide, 7.5 million people in the United States. It's an autoimmune disorder. It does have a significant lifestyle and psychological impact on patients. Again, this is a systemic and local autoimmune-mediated inflammation disorder. So using a mocha-mold mouse model, we were able to show a significant improvement with a single administration for mild to moderate psoriasis. We were also able to show with severe psoriasis a significant improvement in PASI scores with multiple administration. We were able to show significant improvement in epidermal thickening and keratinocyte proliferation, significant reduction in that, and we were able to show a decreased immune cell infiltration for both T cells and macrophages. We also compared it against IL-23 monoclonal antibody, which is a product that is approved for the treatment of psoriasis. We were able to show that a single administration of our product was just as good as multiple administration of the monoclonal antibody against IL-23. Looking at local impact on cytokines that are pro-inflammatory, such as IL-23, TNF-alpha, and IL-12P70. We were able to show significant reduction in these lesions for these inflammatory cytokines. And incidentally, all of those cytokines are the ones that are impacted currently by Cosentyx, Trimefa, and Humira, which are products that are marketed and approved for the use of treatment of psoriasis. We were also able to show that administering fibroblast intravenously does not cause any fibrotic response, and we were able to show that with procollagen 3 and terminal propeptide analysis. So in summary, significant improvement of moderate to severe psoriasis using a single and multiple administration of the spheroids administered intravenously, and no adverse side effects noted to date. We are a clinical stage company with pipeline ready to proceed to clinical trials. We have a very strong IP platform with 220 patents. We're very open to academic and industry collaboration, and we are beginning a clinical trial for diabetic foot ulcer in the second half of this year. Thank you very much.